Hello, everyone, and thank you to the organizers for the kind invitation. I'm very honored to join this ACPSB meeting. Unfortunately, I cannot be there in person, so I'll be giving my talk online today. I'd like to share with you a fascinating plant that, in my opinion, has a history of over 2,000 years, pyrethrum. I'll talk about its unique and surprising interaction with insects. As we know, plants have evolved a wide variety of secondary metabolites to manage their interaction with insects. Among these, plant volatile play crucial roles as signaling molecules, attracting pollinators, spreading seeds, repelling pests, attracting natural enemies of herbivores, and even affecting soil microbes and pathogens. In nature, plants face a challenge. They must defend themselves from pests while still ensuring successful reproduction. For flowering plants that depend on insect pollination, it's not easy to do both. Traditionally, plant defense and pollination were studied separately, but now we need to consider how plants balance these two processes and rethink the plant-insect relationship. So I will introduce a special plant in the picture. Pyrethrum is a perennial herb from the Asteraceae family. It shows strong resistance to aphids and has been used as a natural insecticide in the West for over 150 years, and that's because its flower heads produce pyrethrins, a type of toping compound. Interestingly, pyrethrum was discovered as far back as 2,800 years ago in the East, where it was called 除虫菊, means kill insect chrysanthemum. Its flower head can contain up to 1.5% pyrethrins. Before World War One, Japan was the largest exporter of pyrethrum. After the war, the UK began looking for the suitable cultivation regions. Due to its ideal climate and geography, Kenya started growing pyrethrum and overtook Japan by 1939 to become the top exporter until the uh, end of World War II. This picture shows a pyrethrum extraction factory in Yunnan in China. Flowers are harvested at stage 4, grounded into powder, and then distilled at large scale. Then essential oils are then refined in the lab and turned into final products. There are many products like mosquito coil, pesticide, and the pyrethrins act as neurotoxins, killing insects on contact. They don't lead to resistance and are environmentally friendly and are harmless to humans and animals. So in the field of Yunnan, farmers often interplant pyrethrum with their fruit trees and vegetables. They told us that once pyrethrum is planted, pests disappeared from the garden. But here is the mystery. Pyrethrum are not volatile, so they stay inside the plant. So how do they affect neighboring plants and reduce pests? So this raised three key questions for us. So first, do any insects still visit such a pest-resistant plant? And the second, if yes, why are they there? And the third, how does pyrethrum protect the nearby crops? So we first surveyed the insects on field-grown pyrethrum in Yunnan. Surprisingly, we found a huge number of ladybird beetles. So nearly 80% of all insects observed were beetles. There could be over 100 sudden beetles per hectare. So even more interesting, we didn't find any aphids, which are their main food. So that made us wonder, why are all these beetles coming to pyrethrum without any food? So and then we think maybe it is because of the volatile. So we collected volatile from beetles attracting and non-attracting pyrethrum genotypes using GCMS. And the key difference was a compound called ebetafinacy. So I will call it EBF later. So it is an aphid alarm pheromone. So aphids release EBF when threatened by predators like beetles to warn others to flee. And we also analyzed the volatile at different flowering stages, so pre-flowering and S0 to S2 early stage, and S2 to S4 blooming stage, and S4 to S6 later stage. So another compound, germacrene D, so GD, was found in high amounts. So interestingly, the ratio of EBF to GD decrease as flower mature. The previous studies suggest that GD can suppress the effect of EBF. Only when GD levels are low and the EBF is pure can it effectively attract beetles and repel aphids. So that's why we saw the most beetles at early flowering stage. Also, when plant is damaged, EBF release increase, suggesting it's also stored inside the plant. So using a Y-tube behavioral assay, we confirmed that beetles prefer flowers at the S2 stage, where EBF is most abundant and pure. And also the EBF standard and damaged flowers attracted even more beetles than undamaged ones. So here we show that EBF can attract more beetles. So since aphids usually feed on the flower stem of plants, and the beetles also like to stay at flower stem, so we separated flower buds and the flower stem, and we found that pure EBF was mainly released from stem. And landing staining, which is staining can show the tauping to blue color. So our landing staining showed tauping compounds in the phloem of the stem with secretory cavities, possibly storing some EBF. And we can see such blue dots here. So, but what's the point of storing so much EBF? And apart from slow release to attract the beetles, we wondered that since aphids feed on the phloem, so could they also ingest EBF? So we fed aphids on pyrethrum stems and we collected their honeydew. So we detected undigested EBF in the honeydew, so mimicking natural alarm release. So this is a very interesting and hard assay. So we have to collect the tiny droplet within half a second using a microcapillary. So to test the effect, we compared aphids raised on cabbage and those raised on pyrethrum and also cabbage with EBF. So we can see that the aphids adapted to pyrethrum were less responsive to flower odor as well as EBF, confirming their desensitization to the compound, even if the concentration of EBF in the honeydew is very low, but it's enough. So we proposed that pyrethrum store EBF in the stem's flow, allowing indirect defense by attracting beetles and direct defense by feeding aphids excrete EBF to scale others. So if you remember, earlier I mentioned GD, so another important compound. So we may curious that what's the function of GD, and Pyrethrum is a flower plant with highly insect resistance. So how this plant pollinates? And interestingly, EBF and GD are both derived from the same precursor, FPP, via different enzymes. So the ratio of EBF to GD changes over the flowering stages, which is quite fascinating. So in the early stage, the EBF is high, and in the blooming stage, the GD is high. So GD has been reported to attract nocturnal insects. So we formed two hypotheses. So EBF mainly functions in defense, while GD might play a role in pollination, especially at peak flowering in the night. So to test this, we carried out pollination experiments in Vienna. So we used cages to cover the pyrethrum and compared the seeds set with different treatment. So for example, the fully enclosed, so no pollinators can go into the cage, and daytime only means only let daytime pollinators go in, and the nighttime only, which means only night pollinators go in and open plots. So results show that nighttime pollination significantly improved the seed yield and the quality. So we also did manual pollination at night and daytime. We found that night also gave better results than daytime pollination. So we think pyrethrum is mainly pollinator at night. So we solved the nocturnal insects on pyrethrum, and we found moths to be the main visitors. So we focused on two moth species, cotton bollworm and pyrethrum, for our lab studies. And then we found that flowers release more GD at night, especially during the S2 to S4 flowering stages. And they have an interesting day and night dynamic emission. So this GD emission comes mainly from receptacle and bracts. So we also performed window tonal experiments. So we can see that the moths prefer S2 to S4 stage flowers, and also GD standard attracted them. So we think pyrethrum may release GD at night to attract moths for pollination. So to understand how GD is produced, we cloned the GDS gene from flower heads. So it has two transcripts, GDS1 and GDS2. So GDS1, when expressed in tobacco, it produces GD, and GDS2 has a splicing error and no activity. So GDS1 is most highly expressed in early flowers. Maybe this is for ready for produce GD for the late stages, and it also highly expressed, especially in breasts, and also matching GD accumulation patterns. So interestingly, unlike other terpenes, the GD and EBF aren't made in glandular trichome. So gut staining show GDS expression near vascular bundles in the cortex, and we wanted to see how this spatial expression affects GD release and moth attraction. So when we observe moth behavior, we noticed that they hovered over the flower receptacle. So GD was most enriched in the bracts, and our landing staining showed that the top of bracts had many terpene staining, and GD might release from here. So we also perform SEM to observe bracts structures. So it showed bracts had many stomata and secretory cavities. So this suggesting that GD is stored and released from these structures. So we also find that moths will stay on disc florets. So we also did landing staining, show that that terpene-rich security secretory cavities in the receptacle. So we observed that GD also reach oil in the corolla and the stigma. And here are two very clear secretory cavities represent the stigma GD storage place. So this suggesting that GD is also released during flower opening. So we also found another very interesting phenomenon. So is that during our moth choice experiments, and we found that moths also lay eggs on S2 to S4 flowers. They laying eggs on flower stems and the leaves. However, most of eggs will not hatch. And this raises a concern. If moths are pests, so after pollination, wouldn't they larvae harm pyrethrum? So we tested larval survival on pyrethrum versus other two chrysanthemum. So larvae of two moths fed on pyrethrum showed slower growth and all died. So this suggests pyrethrum is not a suitable host for moth larvae. And we then suspected that this is because of the toxin of pyrethrums in the pyrethrum. So we sprayed different concentration of pyrethrum on regular chrysanthemum leaves, and larvae died within 2 to 3 days. So here is our conclusions. So pyrethrum uses a clever two-phase strategy. So one phase is defense, another phase is pollination. So during the daytime, so it releases EBF and to attract beetles and repel aphids for this defense. And at night, it releases GD to attract moths for pollination. So although some moths lay eggs and few hatch, and their larvae cannot survive due to the plant's presence. And also at night, because of the pollination, so the seeds yield is higher than the daytime seed yield. So this studies shows how a plant can skillfully coordinate its secondary multipliers in time and space to solve the conflict between defense and pollinations. So that's why I call the pyrethrum is a very clever plant. And last, I'd like to thank Professor Wang Caiyun and Professor Wang Manqun, Chang Xiangqian, and Dr. Li Jingjing, and other colleagues from Huazhong Agricultural University, and also thanks to Professor Martin Jansma in Wageningen University for the help. And also thanks to Nambao Company in Vietnam for field support. And this work was funded by the National Natural Science Foundation. And also, thank you very much for your attention, and I'm very happy to take any questions.